Let me see. Is this on? Okay. So thank you everyone for joining us for the Cancer Center seminar today. I'm very excited to welcome Dr. Leif Bergsagel. He is a professor of medicine for the Mayo Clinic College and is a consultant for the Division of Hematology and Medical Oncology in the Department of Internal Medicine at the Mayo Clinic in Arizona. He is the principal investigator of the Mayo Clinic Multiple Myeloma SPORE program, which is a translational cancer research program that aims to understand the genetic and biological mechanisms driving myeloma and has received funding from the National Cancer Institute. Dr. Bergsagel received his bachelor's degree and his medical degree from the University of Toronto. From there, he completed residencies at the University of Toronto Sunnybrook Medical Center, as well as at Stanford University Medical Center, before completing a fellowship program in medical oncology at the National Cancer Institute in the laboratory of Dr. Michael Kuhl. After working at Weill Cornell Medical College in New York City for 8 years, he moved to the Mayo Clinic in Arizona in 2004. Dr. Bergsagel has made numerous discoveries that have substantially contributed to our understanding of myeloma pathogenesis. Working together with his wife, Dr. Marta Chessy, he identified the recurrent chromosomal translocations in multiple myeloma, an array of mutations that activate NF-kappa B pathway, and developed a novel transgenic mouse model that faithfully reproduces the genetic, phenotypic, clinical, and therapeutic features of human myeloma. As an active investigator in many clinical trials, he has led efforts to develop targeted therapy for multiple myeloma, starting with the translocation T414 that he identified. Dr. Bergsagel has won numerous awards, to name a few: the Robert A. Kyle Mayo Clinic Distinguished Investigator Award from Mayo Clinic, Arizona, the Daniel E. Bergsagel Visiting Professor Award from the Ontario Cancer Institute, and the McCardle Lectureship Award from the University of Wisconsin. And actually, sorry, I forgot to grab it. We would like to add to that. And we would like to present you with the Cancer Center's Cancer Warrior Award for all of the amazing contributions you've made in the field of myeloma. I can get that out. There you go.
Thank you very much.
We look forward to your talk. Thank you.
Well, thank you, Lorraine, for that very nice introduction. And thank you, Dr. Charbonneau and students, for inviting me to present here today. I'm going to talk to you about the molecular pathogenesis of human multiple myeloma in the very first part of my talk, and show you how we've modeled that in a mouse model that we call the VK-MIC model in the second quarter of the talk. And in the third quarter, I'll talk about how we've used that to study small molecules to improve treatment of patients with multiple myeloma. And in the final quarter, about how we've used this model to study bispecific antibodies in the treatment of multiple myeloma. But first, I'd like to advance the slides to tell you about the pathogenesis of human multiple myeloma, which is a plasma cell malignancy. And to remind you about the differentiation steps in B cell development, a pro-B cell undergoes VDJ recombination in the bone marrow and then can enter the circulation where it may then go on to enter a germinal center, undergo repeated rounds of somatic hypermutation, followed by isotype switch recombination, and then may home to the bone marrow as a long-lived plasma cell. And so when we looked at this pathogenesis, Dr. Keel and I came to the idea that it was very likely that isotype switch recombination may be important in the pathogenesis of myeloma, because you almost never see IgM myeloma. It's always isotype switch or light chain only. And we set out to develop an assay to look for that. And I think everyone here should be reminded about the history of Southern blots. And so I'm proud to show you a Southern blot. And this was one that I did back in 1995. And this shows you the normal organization of the immunoglobulin heavy chain locus. Oh. Oh, yeah.
One more down. Yeah, that one. Sorry.
Thank you. So this shows you the immunoglobulin heavy chain locus from humans. And I've diagrammed a case where there's been an isotype switch recombination to gamma-2. And what happens is this region in between is deleted. And if you do a southern blot with probes, 5' of switch mu and 3' of switch gamma, you see different kinds of bands. Basically, you'll see one band here, 5', 3' switch gamma, corresponding to the germline IgG4. You'll see a rearranged band, 5' switch mu to 3' switch gamma, showing you the productive rearrangement to IgG2. And then, in this case, you see two bands that are off by themselves, and they don't hybridize to any other probe from the IgH locus. And to figure out what those were, I made a genomic phage library and screened it with these probes, isolated those fragments, and you can map where pieces of the genome are using radiation hybrids. And I was able to map these regions of the genome to 4p16. And that turned out to be the chromosome 414 translocation. As diagrammed here. And this was the first demonstration of that actually in a myeloma cell line, and we identified eventually the genes located at that translocation breakpoint. And we did this for a number of other translocation breakpoints in multiple myeloma. And summarizing many years of work, we identified translocations that affected the expression of cyclin D genes, most commonly cyclin D1 on 11q13, but also cyclin D3 on 6p21. Translocations to MAF, most commonly 1416, dysregulating CMAF, and the 414 that I just told you about, where there's two genes, FGFR3 or MMSET, or as I called it, and it's now called NSD2. And the rest of myeloma is characterized by hyperdiploidy. And I'll get onto that in the next slide. But the events I'm showing you here seem to be the primary events that lead to the immortalization of the clone in multiple myeloma. But that same clone is present in the pre-malignant precursor conditions that we call monoclonal gammopathy of undetermined significance and smoldering multiple myeloma. But if you ask, so what are the features of the disease that never change, that from the beginning, from the very first time you're diagnosed with multiple myeloma, through when you've got relapse disease, through to when you have refractory disease? So Caleb Stein, who's actually here in the audience today, determined what are the features that never change, using both copy number as well as expression, and then asked if you then take those features that are unchanging and say what are the determinants of the differences between the different patients with myeloma, you could see that it fell into 6 groups of patient subtypes. And the subtypes corresponded with the presence of the chromosome translocations in the cyclin D, the MAF and MMSET, and then with the presence of the hyperdiploidy. And so we think this forms, I don't know how to get rid of this bar. So I'll try that again. This forms the basis for an invariant classification of multiple myeloma. So if you then take those primary events and look at a series of patient samples, this is from the COMPASS data set of the Multiple Myeloma Research Foundation, and these pinwheels show you the expression of cyclin D1 or 3 in the patients with the 1114 translocation and the few with 1214 cyclin D2 translocations, or the MAF translocations, or the 414 chromosome translocation with expression of NSD2 and FGFR3. And then if you then use the chromosomes that are frequently trisomics, 5, 9, 15, 19, and 11, You can identify a classification based on the presence of the hyperdiploidy with and without chromosome 11, and then a set of patients that have neither recurrent chromosome translocations or hyperdiploidy. And that corresponds exactly to the groups that I showed you previously. And these groups have characteristic secondary genetic events, which can be gains and losses of other chromosomes. A frequent adverse one is the gain of 1q and the loss of 1p. And a distinct pattern of mutations that vary based somewhat on the primary genetic event. So we think basically that multiple myeloma is 6 different diseases and that they follow slightly different paths and may respond slightly differently to treatment. And I'll note here among the hyperdiploid patients that they have the highest level of MYC translocations. About 50 or more than 50% of them have MYC translocations. That'll become relevant when we get to the mouse. To sort of summarize that, we have the primary events, and then we have this variety of secondary events, most commonly the RAS mutations, NF-kappa B mutations, and MYC rearrangements. And there's also phenotypic differences between the early stages and late stages, not just genetic differences. And so this slide shows you on the bottom how much of the transcriptome of the cell is devoted to immunoglobulin transcription for normal plasma cells, a few patients here that have MGUS, newly diagnosed multiple myeloma, myeloma samples taken from patients in minimal residual disease, relapse refractory myeloma, and then the myeloma cell lines we use for all our studies. And you'll see actually how different the myeloma cell lines are we use from patients with newly diagnosed multiple myeloma. And on this axis, you're seeing a measure of gene proliferation. And basically, as the cell makes less immunoglobulin, it's more proliferative. So there's a sort of a counterplay between proliferation and immunoglobulin transcription. And to try and give you a model to put these genetic events that you can sort of understand what's happening biologically, I'll just give you this sort of short description. That when the cells have got a MYC translocation, these are patients from the COMPASS data set with MYC translocation, there's no correlation between activation of NF-kappa B as measured by this index. And actually, patients that have got activating mutations of NF-kappa B are labeled here in yellow, and the level of MYC. They seem to be totally independent. But if you look at patients that don't have a MYC translocation, then you see a very sort of linear correlation between the level of NF-kappa B, and here you see the patients with the mutation that are higher, and the MYC translocation. And these patients down here tend to have actually inactivation of MAX, a negative regulator of MYC. And so the model that I'd like to present to you for the pathogenesis of myeloma is that a normal plasma cell homes to the bone marrow, and it secretes soluble BCMA. It gets cleaved by gamma secretase. And by secreting BCMA in the environment, I think it prevents other factors, other plasma cells from honing in on its niche. So it's protecting itself by secreting BCMA and keeping all of the BAFF and APRIL ligands for itself. But what the BCMA does is it signals through NF-kappa B, and that leads to some MYC upregulation, as I showed you in those patients. But that in order to expand its plasma cell niche from a normal plasma cell or MGUS from 5% to 25%, it needs to find some way, either stimulating the tumor microenvironment to make more BAFF and APRIL, or find ways to constitutively activate NF-kappa B with different mutations, or to go all the way downstream and dysregulate MYC or inactivate MAX. And leading to the upregulation of, and if you look at it, 42% of the time there's a MYC translocation, and 21% of the time there's an NF-kappa B mutation. Other times we find that there's an upregulated NF-kappa B index without mutation, and about 4% of the time there's MAX inactivation. And those patients that don't have any one of these frequently have RAS mutations, and only about 6% of patients are sort of unresolved in this model. Summarize the first quarter of the talk. The VK-MYC mouse model, oh, I've put the wrong slide here. Sorry. I'll summarize it with that. The VK-MYC mouse model models the events that we see in human, the genetics of human multiple myeloma are centered around dysregulation of MYC, which can be achieved either through MYC translocation, NF-kappa B activation, or trans-acting factors. So we set out to model what we thought was one of the most important events in multiple myeloma, the activation of MYC. And we created a transgene by inserting into a construct that mimicked the normal kappa locus, the exons 2 and 3 of human MYC. But here we replaced the third codon with a stop codon. And so what happens in these, if we make a mouse where we didn't, and we made a mouse where the third codon was not mutated, and those mice developed probe lymphoma and died within 30 days. But when we mutated that third codon, there was transcription of MYC message, but there was no protein. And so there was no phenotype. But we put this stop codon in the framework that it could be targeted by somatic hypermutation in the germinal center. And sporadically, an individual B cell would revert that stop codon and begin to translate MYC. It turns out that those mice develop multiple myeloma. And what we see is MYC protein expression in plasma cells, but they're not proliferative, which is really the phenotype of human multiple myeloma. And you see plasma cells accumulating in the bone marrow, not the spleen. And you see a monoclonal serum protein, an M spike, developing starting around 30 weeks and progressing over time to become large. And these phenomenon is more frequent in males. We analyzed 2,600 mice. And you'll see that as in humans, there's slightly faster incidence of multiple myeloma in males as compared to females. And we think that this event, the MYC activation, is not the primary event in this case. We think it's the secondary event, much as it is in human multiple myeloma. And that's based on this experiment where we introduced the transgene into BALB/C mice. And in wild-type black 6 mice develop a monoclonal gammopathy, but the wild-type BALB/C mice do not. And when we put the transgene in the black 6, we get the multiple myeloma I just showed you, but we do not see that in the BALB/C. And so what we believe is happening is that there's a spontaneous monoclonal gammopathy that gets accelerated by the MYC. And we see the malignant features of human multiple myeloma. We see anemia, we see protein deposition in the kidneys, we see osteoporosis and occasional lytic lesions. And if we look to see, are the immunoglobulin genes somatically hypermutated, as in human myeloma? This is a measure of the number of mutations in the V gene in human myeloma and in the VK-MYC model, and actually in the BALB/C plasmacytomas. And this is lower than what's seen in Klaus Rajewski published this year, a new model of multiple myeloma based on turning on cyclin D1 and IKK2 or NSD2 and IKK2 using IgG1 Cre. And what happens there is you turn on the Cre, you turn on the NF-kappa B quite early, and the cells are forced to exit the germinal center. And so they do not get many somatic mutations. And the cells are isotype switched. And so using this, we've developed 3 models of VK-MYC myeloma. There's the model that I've just told you about of aging the mice to about 70 weeks. We call that the de novo model. And this slow course allows for the spontaneous acquisition of progression events. And this is in contrast to other models where you put in multiple oncogenes and the tumor develops quite rapidly in the mouse, but they don't have the diversity of mutations that you see in human disease. And from this de novo model, we have done next generation sequencing. Sometimes we're able to get the cells to grow in vitro and form cell lines, about 1.5% of tumors. And then we've taken them and we can transplant them into syngeneic hosts, so black 6 mice that don't require any pretreatment, and they typically engraft in about 4 weeks. And we call this the transplant model. And so we have developed 24 different cell lines that grow in vitro and 58 different cell lines that are transplantable from one mouse to the other. And to show you that the mouse looks very much like the human, I'm showing you again immunoglobulin transcription versus proliferation for normal plasma cells, and in fact Klaus Rajewski's model here of cyclin D1 and NSD2 with IKK, and then the de novo mice and the transplant mice and the cell lines that grow in vitro. And here again is the effect on proliferation, looking very much like the progression that we see in human disease. And if we look at just the total number of mutations using whole genome sequencing, we see that the number of non-synonymous mutations in de novo mouse model is similar to what we see in patients with smoldering multiple myeloma and stable disease. And what we see in human myeloma and in the smoldering myeloma that progresses is similar to the number of mutations we see in the transplant model. And then in the in vitro cell lines, we see more mutations still. So they're really the same number of non-synonymous mutations. And this becomes important when you start thinking about immunotherapy and the neoantigens that might be present. We also see signatures of different genetic events that occur. And these are based on a whole genome sequencing. And one signature that's been shown to be important in human multiple myeloma is an ApoBec signature. This shows you patients with multiple myeloma where different signatures have been identified. And this ApoBec signature has been associated with those patients with smoldering multiple myeloma that end up progressing to myeloma, and is not detected in those patients with stable smoldering multiple myeloma that do not progress. And so we wonder whether this ApoBec signature might be involved in causing the progression. And when we look at our mouse model, we see the same feature. We see this ApoBec signature present in a fraction of the multiple myeloma that we have. And so one of the experiments we plan to do is to see if we introduce ectopic ApoBec expression, whether we can accelerate the progression of the disease. And is this ApoBec signature that we're seeing in humans, is it driving the process, or is it perhaps just responding in some kind of bystander effect? To give you a picture of the genomic diversity of multiple myeloma, I'm showing you just the copy number changes in de novo mice that we've analyzed, the transplant model, and the cell lines. And the picture I'd like you to take home is that there are frequently gains and losses of chromosome, much like what we see in hyperdiploid multiple myeloma, with some regions synthetic to human regions. We see a loss of chromosome 13 that sometimes includes RB, 17p that seems focused on NCOR1, a 1q synthetic region that contains MCL1. And if we look then at those panel of cell lines that I showed you, in which we've analyzed them by whole genome sequencing and transcription profiling, we see a variety of different mutations. These are all 2 de novo, several transplant, several in vitro cell lines here. And you'll see a variety of different mutations, but with focus, as we see in human multiple myeloma, especially towards the end stages of cell cycle and p53 dysregulation. And we see multiple different mutations that converge on NF-kappa B, similar to what we see in human multiple myeloma. And to highlight one in particular, MAP3K14 is actually the most common IGH translocation. That's not one of the primary. It's about 5 to 8% of patients with human myeloma that got an IGH translocation to MAP3K14. And we identify that translocation actually to IgK as well as to IgH in the mouse model. And those 2 samples have very high levels of MAP3K14 transcription. But you'll see that there's other samples here that have similarly high level and of what looks like outlier expression. So we went on to study it further. And what we identified was an intracisternal A particle insertion. So that's an endogenous retrotransposon that is known to be active, actually, particularly in mouse plasma cell tumors. And this shows you the RNA transcription. This is a control transcription where you see exon 2 and exon 3. These are read counts. And different insertions, and the insertion site is typically about 6 to 8 nucleotides long, and it shows up as a little blip here. And those are the insertion sites of different retrotransposons into the first intron, or actually here into the exon. And these mutations abrogate the binding of BRC2 and 3, which ubiquitinate CMAF and lead to its degradation. So they not only upregulate the expression but of the transcript, but they upregulate expression of the protein by abrogating the signal for degradation. And if you look then, this again is the transcription of MAP3K14. These are in gray are all the samples with the IAP insertion, the 2 immunoglobulin translocations in red, and then a variety of other factors, which have got a high NF-kappa B index and mutations of those genes. And finally, there's mutations that converge on the RAS pathway. And so there was one mutation of NRAS and one of KRAS, but also several mutations of PTEN. Now you might ask me, why do I put PTEN into the RAS pathway? And that's based on this paper from Ryan Young from the NCI, who showed in plasma cell tumors that the RAS can co-opt mTOR and the amino acid sensing machinery, leading to PI3K activation. So it's important in the plasma cells, which all that level of immunoglobulin transcription, it's important that they have the amino acids they need for the production. And we think in this case that the PTEN in the mouse is serving the same purpose as the RAS mutations in the human. And we see both biallelic deletions as shown here by the copy number plot, but also a variety of mutations in the first 1 or 2 kb downstream from the initiation of transcription. And these mutations preferentially target T. I think 27 of the 40 mutations were T and A, as compared to G and C. So it's not an AID driven process which targets G and C, but it's an A to T mutational process driving the PTEN mutations. And finally, the last set of mutations are mutations affecting chromatin modifiers. The KDM6A is known to be commonly mutated in human multiple myeloma, as are other ones. But the most common one here was NCOR1. And sometimes it was inactivated by IAP LTR insertion, as I showed you for the IAPs can not only turn on genes, but they can also turn off genes. And so here in this case, there was an insertion into exon 39. And in this case, there was an insertion here into between exons 21 and 22. And the transcript going from 22 up is known to be a dominant negative of NCOR1, so a way of inactivating NCOR. There were also biallelic deletions and nonsense mutations. But the importance of NCOR1 was recently in myeloma was shown by Ricky Johnstone in this paper where he looked at P300 inhibition. But he showed that by knocking down NCOR1 with sgRNA, that led to marked upregulation in MYC and IRF4 that we know are critical regulators in myeloma. So with that, I want you to leave you with the thought about P300, and I'm going to conclude this section, which is that MYC dysregulation drives the progression of monoclonal gammopathy to myeloma. And because it's indolent, we see spontaneous progression events in tumor diversity. And we've got multiple cell lines, some of which are human cereblon positive, which I'll get to later. And the progression is associated with increased proliferation and decreased immunoglobulin secretion. And the landscape correlates with what we see in human myeloma with activation of NF-kappa B, RAS, TORC pathway, and chromatin modifiers. So I'm not going to talk to you about using the mouse model to treat multiple myeloma. And this shows you a seroprotein electrophoresis done on a mouse. And the monoclonal immunoglobulin that this mouse makes is shown here, and we measure it at day 0. And then we treat for 2 weeks, and we measure the M spike at the end of treatment. And here we used, this was treatment with bortezomib. And we show that the spike at this point, compared to the spike at this point, it's 11%. And we plot that on a graph like this. So every dot you see here is a different mouse treated with a different drug for 2 weeks. And we plotted the M spike at day 14 compared to the M spike at day 0. And over here, I've identified drugs that show what I would call a positive response rate, meaning about more than 20% of the mice had a response to the treatment. And you can see many drugs that are in blue are drugs that have shown activity or have been approved for use in the treatment of patients. And then over here, you'll see this is only a small number of the drugs we tested, but these are drugs that showed activity in cell lines, they showed activity in xenografts, but when you put them in this model, they don't show activity. And one drug I'd like to point out here is IMID. IMIDs were not active in this model, and we know that, in fact, mice don't respond to IMIDs. But so if a drug didn't work in this model, 92% of the time, it was never approved for use in myeloma. And if it did work in this model, about 70% of the time, it was approved for use. So let me get to IMIDs. The thalidomide was first marketed in the 50s in Germany, and it was not thought to cause teratogenic effects because the preclinical studies were done in rodents. But it obviously showed out to cause phocomelia, as shown here. And it was in 1990s that Judah Folkman discovered that it had anti-angiogenic properties in rabbits, and that eventually led to its use as an agent in multiple myeloma, where it was found to be active. We now know the mechanism of action of IMIDs, and we know why they don't work in mice. But IMIDs bind to cereblon, and they're one of the first molecular glues that leads to they attract Ikaros and Aiolos, and it gets ubiquitinated and degraded. Mice have got a mutation or a difference in the cereblon gene, and the rodent cereblon does not bind IMIDs. But in the loss of Ikaros and Aiolos, we see a decrease in IRF4 and MYC, decrease in TNF, but an increase in IL-2, leading to T cell activation. So to have a mouse model responsive to IMIDs, we generated 2 transgenic mice using BACs. And one BAC had a clone that really just included cereblon locus, and the other BAC that we've used more extensively included the interleukin-5 receptor alpha gene in more regulatory regions. And with those mice, we showed that if we treated with cereblon, here we treated with iberdomide, at 6 hours, we'd see decrease in Ikaros and Aiolos, but this mouse had a relatively low level of cereblon. And this mouse with a high level of cereblon had much better degradation of Ikaros and Aiolos. And we've used this mouse for most of the future studies. And we saw that this leads to an increase in, or a decrease rather, in T cells as well. So now with this new mouse model, we actually have 100% negative predictive value. We haven't found a drug that works in people, but doesn't work in the mouse. And we've tested many more drugs than this. I'm just showing you a fraction of them. And we've gone on to test additional agents having this. So we've tested bispecific antibodies work great, selinexor. See, anything else I want to point out here? Here, IMIDs are now active in this model. Some drugs that are active in this model are not active in people. Paraphosine is one, and that basically is kind of a dirty kinase inhibitor and is very toxic in people. The histone deacetylase inhibitors have been approved for use, but they've been withdrawn, I think, in large part because of their toxicity. And the fact of the matter is mice can handle higher doses than people, and they don't complain and they're compliant. So it's understandable perhaps why the positive predictive value is only 75%. But so what about some of the new drugs? So this shows you some new drugs we've tested in this model. And I'm going to talk to you about a P300 inhibitor, but also we used a pyrophosphoplatinum which actually is quite active in this model and had some promising activity in the phase 1 study we did at the Mayo Clinic. LCL-161, which is a IAP antagonist, which when we combined it with cyclophosphamide, had a 10-month progression-free survival, which wasn't bad, but is not being developed further by Novartis. Melflufen, a new form of melphalan. This is another kind of alkylating agent. Bromodomain inhibitors, of which we tested a number, all showed activity. And so I'm going to go on and talk to you about the P300. So myeloma, I think, as I talked to you earlier, is an enhancer-driven disease. And we have factors, Ikaros and Aiolos, IRF4, NF-kappa B, that bind to the super enhancers, which is often the immunoglobulin super enhancer, but also tissue-specific super enhancers, such as the one that regulates IRF4. And there are important co-activator proteins at the enhancer sites, such as P300, and also the targets of the BRD4 inhibitors. And the drugs that we use to treat multiple myeloma, there's a classic combination that we use, which we call VRD, for Velcade or bortezomib, Revlimid or lenalidomide, and dexamethasone, all target factors which are at the enhancers in myeloma. The IMIDs lead to degradation of Ikaros and Aiolos, the glucocorticoids displace NF-kappa B, and the proteasome inhibitors inhibit the processing of NF-kappa B. And we postulate that if we could combine those with a P300 inhibition, we'd be able to focus the activity of that co-activator only on the area of that are important for myeloma. So this shows you if we treat a large panel of human myeloma cell lines with pomalidomide, and we measure the change in proliferation, which is really, you don't see death in vitro with these drugs, you just see a decrease in proliferation. You can see some cell lines show a decrease in proliferations, many do not, and some actually have a slight increase in proliferation. And we can correlate that with changes in the proteins. And so you can see Ikaros and Aiolos almost always go down. This shows you the decrease from day 0. So there's usually more than a 50% reduction in the levels of those. But that doesn't really correlate with the change in proliferation, which is shown for you here. The Ikaros goes down, but the cells still proliferate at about the same level. The one that correlates the best is the change in MYC, and also to some extent, the change in IRF4. And shown for you in green is the sensitive cell line KMS11, and then in red, the resistant cell line RPMI8226. And so in order to increase the efficacy of IMIDs, we looked at the combination with a P300 inhibitor. This is a compound GNE781 from Genentech. And these are showing you a synergy plots with increasing concentrations of pomalidomide on the x-axis and the GNE on the y-axis. And you can see in these two cell lines in particular, you see marked synergy, but also some synergy in the others. We did the same thing with a BRD4 inhibitor, as well as with dexamethasone to show you some contrast. And when we look at the change in the protein level, we can see marked decrease when we use the combination versus either one alone for both MYC as well as IRF4, particularly for some cell lines such as this one here in blue, which was resistant. We see it goes down nicely, IRF4. And correlating with that, we see a decrease in viability. And when using our mouse model of multiple myeloma, we treated the mice for days 1 to 5 and 8 to 12, and then stopped treatment. And we looked at their overall survival, whether they were treated with vehicle in red or pomalidomide and dexamethasone. Really no difference with that short treatment. Remarkably, that short treatment with the P300 inhibitor did extend survival, although not quite statistically significant. But the marked effect that you see here with combination of pomalidomide and GNE781 at 15 mg per kg or at a lower dose, 3 mg per kg, these mice appeared to be cured. We waited for a year and the tumor didn't come back, and only with 10 days of treatment. a very remarkable effect. And as a result, there's an ongoing clinical trial looking at the combination of this P300 inhibitor with pomalidomide. And if we look then at this panel of cell lines, each block is ordered based on the expression of the genes shown in that block. They tend to have very high levels of MYC and IRF4 and varying levels of the transcription cofactors. They mostly have high levels of Ikaros and Aiolos, but interestingly, there's two cell lines that are resistant to IMIDs, shown in red here. They don't express Ikaros, but instead they express Helios, and I forget the other one, IKZF2 and 4. And then we looked at the partners that heterodimerize with IRF4, and they're ETS family factors and AP1 family factors. And you can see a similar, or you can see a sort of a mutually exclusive expression of these factors. And I'm showing you selected sensitive cell lines in green and resistant cell lines in red. So there tends to be increase of these transcription factors in the cell lines that are resistant. And here I'm showing you the same thing for the AP1 factors. And particularly, if you look at the BADF, BADF2, and BADF3, you see very nice mutual exclusive expression, again, then with FOS, and then some that seem to have low levels of those. And so our colleague, Dr. Nizar Bailas at University of Calgary, has studied the role of ETV4, but I'm going to tell you about the role of the BADF family transcription factors. So here we took a cell line that is resistant, KMS12, where is it? Here. And we knocked down BADF, and we showed that we made it more sensitive. And then we took 4 different cell lines that are sensitive to the combination of pomalidomide and the P300 inhibition, and we introduced BADF, and the introduction of BADF in all 3 cases made them resistant. So we think that the ectopic transcription factor can mediate resistance to IMIDs. And in these 2 cell lines, we introduced a BADF, which has got a mutation, which is known to inhibit its heterodimerization with IRF4. So it can't heterodimerize. And if it can't heterodimerize with IRF4, it is unable to successfully rescue the cells from the combination treatment with the IMID and P300 inhibitor. And we can show this not only for BADF, but also for BADF2 and for BADF3. And what we see is that in the presence of BADF, the IRF4 expression is maintained. And so we think basically the reason the IMIDs and IRF POM300 combination doesn't work is that IRF4 transcription is maintained. So to change gears now, I'd like to say that even though the cells, started with a MYC translocation or MYC activation, we were able to show that even at the late stages, they still remain dependent on that event. And that's because when we made the mouse, we put LOX-P sites on either inside of the kappa enhancer and crossed the mice with the mice expressing a tamoxifen-inducible Cre. And we made mice that didn't have LOX-P sites around the enhancer and also crossed it with a tamoxifen-inducible Cre. And then we treated with tamoxifen and removed the enhancer driving MYC expression, which gets rid of MYC expression. And what we see is that when there's no LOX-P sites, the tamoxifen doesn't change the expression of the M spike. But in the case where we remove MYC expression, the cells, the M spike goes away. So the cells remain dependent on a primary event, on MYC, even late in the course of the disease. So to finish this section of the talk, the model has got remarkable therapeutic fidelity. The drugs we use to treat myeloma target transcription factors at super enhancers, and targeting either P300 or BET with super enhancers is effective, but they have a very low therapeutic index because those drugs target all the enhancers. But if we combine them with IMIDs, which focus the activity on myeloma super enhancers, they're very effective and a much better therapeutic index. And the resistance to IMIDs, as well as the combination, can be mediated by ectopic expression of the IRF4 heterodimerization partners of the ETS or AP1 family. And despite the accumulation of these progression events, myeloma remains dependent on MYC expression. And so for the final part of the talk, I'm going to talk to you about using this to study bispecific antibodies. So BCMA-targeted T-cell therapy has changed the landscape of the treatment of myeloma, and there's been 2 CAR T-cells approved, and these have a really high response rate, complete response rate, although they do not appear to be curative. The patients tend to relapse, but after some time. And a bispecific antibody targeting BCMA has been approved with slightly lower response rates and a PFS of about 1 year. And in order to understand how to use these better, without having to use a lot of patients, we need an immunocompetent orthotopic preclinical model like the VK-MYC model. So we obtained from BMS and from Pfizer bispecific antibodies that are murine surrogates. So they recognize murine BCMA and murine CD3. And if we take old mice and we treat them with the bispecific antibody, you can see before treatment, the M spike is increasing. And then when we treat, the M spike comes down, and we treat just 2 doses here, day 1, day 8, and then it will come back. And you see the one with the highest tumor burden at treatment is the one that comes back fastest. And in the transplantable model, which is the same tumor transplanted into multiple recipients, we see that if we initiate treatment when the M spike level is higher, that the tumors don't respond, and those with the lower tumor burden do. And we've gone on to confirm this using other myeloma cell lines that we have. This is the M spike level of the responders versus the non-responders. In all 3 cases, this is a reproducible phenomenon. And we actually had a hard time when we first submitted this paper because the reviewers said, oh, there's no evidence in humans that tumor burden affects response to bispecifics. The reason there was no evidence in humans is that no one had looked. I mean, they didn't have the data. So sometimes it's hard. If you wait and try and publish something before the human experience, they say, well, how do you know that happens in humans? And if you wait and do it after, they say, well, we don't need it anymore because we already did the study in humans and we know what happens. So we did this study first and it was published. And then the human data came out, and the human data shows exactly the same thing, that in the patients that with the high levels of BCMA as a surrogate for tumor marker are the ones that don't respond to these 2 different bispecifics. And we also have some cell lines that just don't respond to the bispecific antibodies, even when we treat them with low tumor burden. And when we look at it, we see that the level of BCMA is higher in the responders and lower in the non-responders. And we plotted out here, frequently the ones with the low level of BCMA have activating mutations of NF-kappa B. But to counteract that, we can inhibit gamma secretase, which cleaves BCMA from the surface. And if we use a gamma secretase inhibitor, we can have high levels of BCMA on the surface of cells. And if we do that in a mouse, we can show that we take a non-responder and the mouse now responds. And so here you can look at high M spike levels that typically would not have responded, that these are baseline levels, are now converted to responders by the using of a gamma secretase inhibitor. And of course, we can try to combine bispecific antibodies with IMIDs, and pomalidomide is the one that we chose. And here, of course, we need to use an IMID-responsive tumor. So it came from the H. cereblon VMIC cross, and we go into an IMID-responsive host that has H. cereblon and treat for 2 weeks. And we see, if we look at the M spike at day 14, the combination is very effective. And if you look at this treatment, you can see the tumor responds, although eventually recurs. And if we look at overall survival, you'll see that the treatment with the combination leads to an improvement in overall survival compared to just the bispecific or just pomalidomide. And this is associated with expression of markers of T-cell exhaustion, as well as T-cell efficacy. But you'll note that by week 2 of treatment, the level of expression of granzyme B goes down. And we think that we're hyperactivating the T cells and they're getting exhausted and losing efficacy. And that's shown here, that we have terminally differentiated T cells, as shown by IRF4 expression at the 2nd week of treatment. So we need to find some way to keep the cells from getting exhausted and maintain their efficacy. So one way to do that would be to add a checkpoint inhibitor. So this is a triple combination now of the bispecific pomalidomide and a checkpoint inhibitor. And that is incredibly effective at reducing tumor burden compared to the treatments singly. And it maintains efficacy. We see a high level of cells that have coenzyme B function. The problem is that it's highly toxic, and half the mice died in the first week of treatment. Mind you, those mice that survive the first week tend to be cured. And if we look at why are they dying, we did a cytokine panel 4 hours post-treatment, and this you see a little bit of cytokine release with the bispecific alone, but when you combine pomalidomide and PD-1, you're seeing very high levels of cytokine release that persists up to 24 hours. So although that's effective, it's basically too toxic for the mice and probably too toxic for people. So then we look to see, well, can we, if we did some pre-treatment, can we improve the efficacy? So I'll show you a series of experiments here where we tried using control bispecific or the real bispecific or lenalidomide and dexamethasone or the combination or pre-treatment with Dex or Lendex. And what we show is basically number 6, the pre-treatment with Lendex followed by the bispecific and Lendex gave you the best outcome with the other ones somewhere in between. And these treatments led to increased T cell activation at the peak response, again, as we would expect. But it does not overcome the T cell exhaustion. So the cells, even with the pre-treatment with Lendex, were exhausted. And we see upregulation of T regulatory cells. So those are both things that we don't want to see. So it doesn't seem like we could find some way to get around this effect of the IMIDs and the bispecifics. And so really what we think is the best combination is cyclophosphamide, where we treat just on the same day as the bispecific antibody. And in this experiment, the mice that received the combination of BCMA and cyclophosphamide had excellent outcome in terms of M-spike levels and prolonged overall survival. And showed that in different tumor lines. And in this tumor line, the mice were re-challenged at day 90, and they did not engraft. So that the short-term treatment with the bispecific and cyclophosphamide led to a long-term protective immunity. And if we look at what happens, the cyclophosphamide is really tempering the T cell activation. And so the T cell number is not affected, but we're not seeing increases in T regulatory cells, and we're maintaining a good CD8 to tumor ratio, and we're maintaining a decent level of interferon gamma effector T cells. And I guess critically, we're not seeing the T cell exhaustion. So we're not seeing the high levels of PD-1, KLRG1, or LAG3 when we use the cyclophosphamide. And we have effective memory shown by TCF1. So to conclude this last part, this mouse model allows us to study the mechanism of resistance and relapse to T cell therapies. And the primary resistance can be high tumor burden or low antigen expression. And we can try and deal with the latter with gamma secretase inhibition. And the combination of bispecific and IMIDs can lead to T cell exhaustion, although it does improve the response. But the combination with cyclophosphamide really improves the primary response, tempers the T cell activation, and leads to long-term better outcomes and cure. And so based on this data, the NCI-CTEP has authorized us to do a clinical trial where the patients will be randomized to receive either iberdomide with teclistamab or cyclophosphamide with teclistamab in a randomized phase 2 setting. And with that, I'd like to thank Dr. Marta Casey. There's a picture of us in front of our laboratory at sunset. The members of our laboratory, Dr. Stein, who I'll mention again, is here today. Seth Welsh, who graduated with his PhD from the University of Colorado. And Aaron Mearmeyer, who has just joined the faculty at Mayo Clinic. We've been supported by the SPORE from the National Cancer Institute, a moonshot grant from the NCI, and we had help from both BMS, Pfizer, and Novartis. Thank you. Yeah.
Thanks for the excellent talk. Fascinating. I was wondering about the bispecific relationship with the tumor burden that you saw in the mice in the clinic. Do you feel like that's a function of too much myeloma or not enough T cells crowded out or microenvironment?
Yeah.
That
That's an excellent question about the T cell response, the lack of, can't even phrase the question. What's the question again?
is it cell problem or
Yeah. I think we don't know if it's the tumor burden inhibits it because it's crowding out the T cells, or is it the soluble BCMA, which is soaking up the antibody, or is it actually the tumor, which is soaking up the antibody? So I, don't have any insight as to what it is, but we do think that T cells in the bone marrow are probably very important. And I guess, I don't know, we haven't tried to design experiments to try and tease. I don't actually know how you would design the experiments to try to tease that out of what it was. But I guess I think the clinical take home would be that now that we're introducing teclistamab to the clinic, try not to do it when they've got too much tumor burden, which is kind of hard because obviously we're doing it in patients who are failing everything else, and they often do have a lot of tumor burden. But you sort of wonder if you can't like cytoreduce them with something first and then give the bispecific. So let's say, you're using DPACE chemotherapy, and then we know you can only use that once or twice and they become resistant, but you use DPACE, and then you come in with the bispecific or anything that you think the tumor might respond to. That's right. That would take a lot of patience to try and also you've got the exact same tumor in all the different hosts. So you really can try that, which I think is why we've got some support from BMS and Pfizer to do these kinds of studies. Yep.
I wanted to thank you for acknowledging the importance of southern blots in science. Great for the sequencer. So with the beautiful models that you showed, do you have any sense of whether or not myeloma keeps recurring despite these amazing different therapies because there's a privileged reservoir somewhere where the cells hide out, or is it just purely intrinsically driven from the tumor being them?
Yeah, you cut right to I think the most important question in multiple myeloma today. How is it possible we have these tremendous therapies and we can have patients with no detectable minimal residual disease for a decade or more, and then it comes back. And so we've got a recent experiment in the mouse that I think maybe is going to help us, but we've treated it repeatedly with bispecific antibody, and a resistant tumor came out. And the resistant tumor had lost all plasma cell phenotype, didn't express IRF4, didn't express BCMA, and we couldn't actually tell what it was. But we sequenced it, and it's got the same VDJ and switch recombination as the myeloma. So it's clearly the myeloma, but it's changed its state. And I think that's what's probably happening. The cell is changing its state somehow, somewhere, and becoming dormant or senescent or a stem cell, I'm not sure what. And that stochastically, it's able to come out of that state and cause the relapse. And so I guess what I'm hoping with this cell line we have now is we can try and stimulate it and see if we can turn it back into a myeloma. And then if we could try and use that stimulus in patients to get the cells out of the dormant, out of whatever they are, out and kill them. All right.
So you said that you've generated some acquired resistance to
bispecific in the mice? Have you tried that with other therapies to see how it changes the disease, like IMIDs or something?
We haven't tried it with IMIDs. And I think don't think we've actually done it with any, it's a frustrating process. Sometimes you can treat the mice with cyclophosphamide and cure them. And some of the therapies are quite effective. And so and then sometimes tumors come back and we don't know if the mice have just developed, because they can get plasmablastic lymphomas, they can get other things. And we can't really tell if the tumor is related to the original one. And we haven't actually gone and sequenced everything, but we did with the bispecific because we had a lot of confidence in it. So we may have, and we just threw them away. I don't know. Or I thought they're probably frozen down. But we haven't, so it would be interesting with IMIDs in particular, because the mechanism of why IMIDs fail in patients isn't, well, I showed you some reasons, but it hasn't been fully explained. Yep.
I'm fascinated by this cyclophosphamide bispecific, sort of thought process. It certainly seemed like within your model that the T cell exhaustion starts to develop really quickly after exposure. What we've been learning about cyclophosphamide in haplotransplant setting, I guess I think you wonder if, just sort of aggregating the actual CRS event, sort of diminishing that T cell activation very early on in exposure might have some long-term effectiveness in terms of the T cell population. I don't know, it just sort of seems like a really unique effect of a chemotherapeutic to sort of shift that T cell, sort of responsiveness.
It does. And if you think about it, obviously the IMiDs and the checkpoint inhibitors just made that worse. So if you had some other way, perhaps it's just that initial event. And if you didn't have that, so that's a good point. Yes.
So it's solid kind of that nuclear receptors like NR4A inhibits the function of T cells. Do you see such cases also in multiple nuclear receptors?
Which nuclear receptors?
NR4A family.
No. I'm not sure actually I'm really familiar with NR4A, but I don't think so. The most important nuclear receptor would be glucocorticoid. And we do see NR3C1 mutations rarely cause resistance in patients. Okay, thank you.